what can cause, what type of mutations you have. You can have several types of mutations that can be caused by different things. You can have spontaneous mutation that will just happen on their own, as we're going to see with the shape of the bases and with the tautomeric forms of the bases. But you can also have induced mutations. Like in the case of Chernobyl, what type of mutation is that? Is induced mutation, because it wouldn't happen if you did not bombard these people with a huge amount of radiation. So that's induced due to either radiation or chemicals or some type of a viral infection will cause mutation in the DNA. Can anybody give me an example of a virus that causes some type of mutation? HIV is the first one that pops up to mind. The HIV will include its sequence of DNA into the genome of the host. You also have herpes does, yes, herpes do that as well. Yes, and then the herpes, you can never quite get rid of it. It's just like HIV in a sense that it gets incorporated into the DNA and it just stays dormant for a long time. But yeah, it causes mutations in the DNA. It gets incorporated into the genome of the host. And several genes, several viruses that were started to be used as gene therapy. For example, adenovirus. It was found that they will also cause DNA mutations, because what happened is that we can I'll briefly touch on the gene therapy here. But what you do is that you get a virus, which is a vector. The viruses are very efficient into putting DNA into cells. So you get this viral vector, and you want to deliver a gene into a part of the body that does not have that gene function. So yes, you can put the gene there. The problem is, usually the genes are expressed in a way that you control. So sometimes you have to increase expression, sometimes you have to shut down the expression. You have to be controlling the level of expression, especially in a gene that is regulated, not the housekeeping gene. Now, if you put a gene in there with a virus vector and you cannot control it, I mean, how good is it? You put it on and it's always on. Maybe it defeats the purpose. Now, there were some types of gene therapy in which the scientists were successful into putting the gene in the vector, in the adenovirus vector, putting it into the person, and having a basal level of expression of a particular protein. But then they found out the person develops leukemia. And they were trying to figure out why. Well, it happens that the adenovirus would get into the bone marrow and would mess up somehow the process of formation of the blood. So you're trying to fix one problem here, but you're causing another problem there. You really have to know how to control and how to avoid these types of secondary problems based on the DNA change, the mutation, DNA damage. Not damage, but change in the sequence. So, okay, so you have spontaneous mutations, and you also have induced mutations. Now, there are two types of mutations that you can have in cells. One type that can happen is in somatic cells. As I mentioned here, we have the little example of this fish that has a mutation on the pigmentation, and is not, if it is not on the gonads, is on a somatic cell, the mutation will make clones of this cell with the mutated sequence as the cell divides. So if the cell is going through mitosis, it will amplify whatever mutation was there. So all the cells that are deriving from this original cell with the mutation, all those cells are going to have the mutation. Because remember, that one is not going to go away. Once it changes, it's changed. And every time the cell divides, it carries the change. Now, if it is in a somatic cell, you're going to see, for example, here, that whatever cells derived are on the fish, and the mutation grew. Like in this case, a tumor would grow. Now, if the mutation happens in the gonads and the gamete cells, then it's going to pass to subsequent generations. Now, this mutation is going to be a permanent mutation that can be hereditary transmitted. So as you see here, if it's in gamete-forming cells, it's going to be transmitted to the offspring. Now, this you can also make sense when you think about, right in the beginning of the class, that we're talking about acquired characteristics. if you have, for example, somebody who lost a finger or the mice who had the tails cut, and then the scientists will breed the mice and all the pups are going to show up with tails. Say, well, I just cut the tail of this mouse. Why the pups are showing up with the tails? Because you cut some somatic cells. You do not cut anything out of the gamete cells. So it makes sense that acquired characteristic does not pass down. Same thing with the mutation in the DNA. If you have a certain tumor and it's localized in a somatic cell, you're not going to pass this tumor to the offspring because it's just not in the gonads. Talking about mutations, as was seen before, when you have alleles that, when you have, for example, recessive, homozygous recessive alleles that are not compatible with life, you have little mutations, so you never get the offspring. You can also have mutations that are called conditional mutations. And those conditional mutations are usually related to temperature. Like, for example, when you have the case of the fly, that when the weather is warmer, it will develop the wings. So that's a type of a conditional phenotype, or the genes of the fly had some type of mutation that will allow for expression only at certain temperatures. Now, also with the conditional mutation, once you know that you can work with conditional mutation of alleles, you can design a particular study of a gene, and you say, okay, this gene is only going to be expressed if I add a certain antibiotic to the cell, or if I add a certain drug to the cell. So you can make a conditional mutant that is going to respond to whatever you want to put in the cell in your experiment. It's a very important tool to have once you go into this field of research. Now, there are some types of mutations that you don't only have one mutation, one event that happens. You have two events that can happen. Like if you have a particular mutation that changed the expression of a protein, and then on the same gene you have another mutation that changes the expression back to the original form. So that's a double mutation in the same protein that reverses the function from abnormal to normal. So when that happens, you say it's an intragenic mutation because it happened in the same gene. First mutation messed up the system, second mutation fixed up the system in the same gene. So that's called an intergenic mutation. Yes?
Sorry, I think you were getting to intergenic mutation.
Yes, that's the next one. So the intergenic mutation is when the first mutation happened in one gene and messed up the system, and then another mutation happened in another gene that has nothing to do with the first gene, but that mutation fixed up the process. So you had the two mutations still, one that messed up, one that fixed it up, but now it's not in the same gene. It's in a gene that had nothing to do with the first one. So the first one stopped the expression of something, and the other one recovered the expression of something. We're going to go into more details on this. But I will show you here. For example, if we have a codon that would be CAA, and I don't know what is the codon for, but let's assume that it's something like leucine. And then you have a mutation that comes in here and changes this C to an A. So now you have a codon that is AAA. That's a mutation that happened. But now that's not leucine. Let's say it is phenylalanine. Just so you know, don't write this down because I don't remember the codons for every single amino acid. So now that this happened, let's say another mutation would happen and would change this, not this amino acid here, but this amino acid here will change it to something else. Let's say ACA, and then this new codon will code again to the leucine of the function that was lost before. So these are two mutations, two events, one here and one there, in the same gene. That the first one messed up the expression of the protein. There was amino acid on that particular protein went from leucine to phenylalanine, and then the other mutation restored the original function, and then you produce your original amino acid again. So those are, this would be intragenic in the same gene. Now, a case of intergenic mutation, what can that be? Something that would happen in one gene, and mutations are going to be permanent. So let's see. So you have a particular protein. Here is your protein. You get amino acid 1, 2, 3, 4. So let's say for this amino acid in here, you have a certain sequence, let's say for example for AAA, that will make phenylalanine. And then you have a certain mutation that will come in here and change one of these here. Give me one codon for a stop sequence. Stop codon. U something. UGA, UGA. So this thing mutated from AGA to UGA. And now this does not code for an amino acid anymore. This is going to code for a stop sequence. So what's going to happen when this protein, when this gene is transcribed, is going to have the mutation in it. Now when it's going to be translated, what is going to happen here, that you have your ribosomal subunits, you have the large and small ribosomes, you have the tRNAs, and then the tRNA that is supposed to be for a particular amino acid came into that part of the mRNA and saw a stop sequence, so it's going to stop. So you have a truncated protein, and you're going to have a loss of function of the protein because it's just a piece of it. It's not going to work anymore. Now, how can you fix the problem? Not with a mutation in this gene, but it could happen that the gene that makes the tRNA and the anticodon part of that tRNA, right there, it could happen that now this anticodon for the original amino acid was what? Okay, AGA is the codon, so that one is going to be TCT. That's the anticodon, right? For the tRNA. So what happens here is that, okay, now UGA, that's a stop signal. Let's say this amino acid is proline. Now, this is proline there, right? This is a tRNA that carries proline. If it happens that the gene that makes that particular tRNA gets a mutation, and now this is not a T anymore, this happens to be an A. Now what's going to happen? That anticodon is not going to recognize AGA anymore. Is it going to recognize what codon? It's going to recognize UGA as the codon for whatever it's carrying, which is the proline, which was the amino acid that was missing there in the first place. So this is an intergenic mutation. This was a mutation in the first gene that caused that first mutation. And then this was a mutation in the second gene, second mutation, that fixed up the problem. And now you get your protein functional as you were expecting if no mutations had happened. Yes?
So what you guys, because now you can't, every time you stop codon, you won't stop.
Yes, eventually. But this is one cell. Okay, this is an example of what an intergenic mutation can be.
Okay, so intragenic means in the same gene. Intergenic means in different genes.